For the purposes of today's PowerPoint, we'll be discussing retinoblastomas, their origins, treatments, diagnostic. For the purposes of today's PowerPoint, we'll be discussing retinoblastomas, their formations, diagnostics, and eventual treatments. Retinoblastomas originate as retinal precursor neuroblasts. These are cells that are present as stem cells for neurons and will eventually divide into ganglions, rods, or cones. Genetics plays a large role in predisposition for this types of cancer. Malfunctions in both copies of the RB1 gene are required in order to cause complete loss of cell cycle restriction, resulting in retinoblastomas, and various tumor locations can arise as a result of genetics. Unilateral tumors are found in non-inherited forms of the cancer and only form in one eye, where bilateral and trilateral tumors form in inherited cases. Bilateral is tumors forming in both eyes, where trilateral tumors are formations in both eyes, as well as the pineal gland or pineal gland in the brain. Different types of retinoblastomas can also arise. Group 1 is a direct result of tumors forming from retinal precursor neuroblasts, and Group 2 is cancers evolving from ganglions, rods, or cones, essentially all differentiated forms of precursor neuroblasts. For the purposes of today, we'll be focusing on Group 1. RB1 is an autosomal dominant trait and can function with only one functioning copy. This is why inherited individuals can form multiple types of cancers outside of retinoblastomas, as well as cancers in both eyes. This is due to all somatic cells in that cell line containing that inherited copy only requiring one mutation instead of the normal two, making it more common. These heterozygous offspring often present as bilateral retinoblastomas and often earlier in age as well. 40% of cases present this way and also contain tumors in other tissues as well, while 60% are non-inherited cases of retinoblastomas. Retinoblastomas occur at a rate of 1 in every 16,000 births approximately in the US, with 11% presenting within the first year of development. And this is an equal split between male and females due to the autosomal traits of the gene. The rate of death is 10% in developed countries. This is due to wide variety of flash photography that occurs nowadays, as well as ease of access to drugs and other forms of treatment. This 10% is usually a result of late-stage diagnoses and other invasions of tissues complicating enucleation, shown here on the left. The diagnosis of retinoblastomas originally begins with the noticing of leukocoria, which is a white pupil, and this is noticed during flash photography or optical exams with a flashlight, and is the visible white tumor at the back of the retina. This warrants further genetic analysis after a visit to the doctor's office, where RB1 is sequenced and directly analyzed for defects, as well as chromosomal 13 Q-arm deletions and methylation analysis to determine the epigenetic condition of both copies to determine predisposition for other cancers. Symptoms of this disease include loss of motor control, causing the eyes to look in different directions, as well as a decrease in the visual field, as well as clarity. This is a result of the tumors directly pressing onto the optic nerves. Irritation and swelling of the eyes has also been noted to occur, but symptoms are very limited in the sense for retinoblastomas, only affecting the eyes and visual fields. However, after metastasis, this can then bring in damage to other tissues and other malfunctions that lead to eventual death. Heterozygous individuals are also at risk due to the bilateral nature of their case to vision and damage in both eyes, resulting in complete blindness. The gene responsible, as I said earlier, is RB1. This is located on chromosome 13, the Q-arm position 14.2. The resulting product is the RB protein. This is a tumor suppressor involved in the restriction of cells to the G0 phase, or can be inhibited in order to allow the progression into G1 and eventual mitosis. Null mutations are required in order to completely inhibit function of a copy, and are usually well conserved and not as susceptible to single or SMP mutations. Heterozygous individuals also possess a larger risk for potential loss of heterozygosity. This is where DNA polymerase will spontaneously jump chromosomes and can potentially copy the malfunctioning copy into the homologous chromosome, allowing for two malfunctioning copies. This can also occur as a form of as a result of non-disjunction and chromosomal recombination during mitosis. RB, as I said earlier, is constantly produced and is a restriction point for the cell cycle. This is because at rest, it is bound to E2F, which is a transcription factor used to encourage and continue the cell cycle into G1 and S phase. After cyclin D is transcribed and binds to CDK4 and 6, RB is phosphorylated by this complex, allowing in the hypophosphorylated state. Once enough RB has been hypophosphorylated, ECDK2 is able to hyperphosphorylate and essentially continue the cell cycle as RB is completely inhibited. This R point is the change from the hypophosphorylated state to the hyperphosphorylated state, allowing for the continuation of the cell cycle. The null mutation eliminates this ability to constantly bind to E2F, allowing it to freely roam and constant proliferation of the cell to occur. This figure right here shows the possible pathways for RB post-hyperphosphorylation. So if not enough CDK is present, and ITF will remove the phosphates, allowing it to be reactivated and continuously bound to E2F, allowing for differentiation to be preferred. If stress is added to the cell during after RB has already been phosphorylated and released E2F, BRCA1 encourages apoptosis and cell death. And this is the pathway for normal cell proliferation, except this is actually showing the inactivation of RB due to SV40 large T antigen, which makes people who have been infected with SV40 and HPV to also be at a large risk for retinoblastoma cancers. E7 would replace the large T antibody for RB in this figure. But this essentially, these antigens would bind to RB, causing it to release E2F and encourage the proliferation of the cell, eventually resulting in retinoblastomas. Treatments for retinoblastoma include cryo and thermotherapies, chemotherapy, radiation, enucleation, and experimental therapies such as specific targeting therapies. Cryo and thermo would involve the use of extreme temperatures to shrink and cauterize surrounding tissue to remove this damaged or affected tissues. Chemotherapy is currently used as a cocktail of called CEV. This contains carboplatin, eptoposide phosphate, and vincristine sulfate. Carboplatin will cause cross-linkage of DNA, resulting in cell death. Etoposide phosphate will result in the excretion of DNA from the cell body and can is the main factor in toxic buildup within the blood. And then vincristine sulfate is a vinca alkaloid that targets the beta tubulin during mitosis, preventing the binding of alpha tubulin, essentially dismantling the microtubules used to separate the chromosomes, and causes apoptosis as a result. once it has gotten to that checkpoint in G2 phase or during M phase. And then experimental treatments such as targeted therapies look for specific proteins active in cancer cells that are not active in normal tissue, allowing for only cancer cells to be affected. This is proving to be slightly difficult with the retinoblastomas due to the proteins involved being constantly on and also used in all other proliferating cells as well. The progression of these tumors is highly affects the prognosis and eventual treatments that will be used as a result, if it is treatable at all. Stage 0 refers to unilateral formations that are still present without the eye being removed. Stage 1 refers to unilateral that has been enucleated with clean margins. Stage 2 refers to unilateral tumor formations that have been enucleated without clean margins. Stage 3 and 4 are divided into A and B. Or 3A describes extra molecular or extraocular, meaning that the tumor has spread to tissues outside of the eye and metastasized, invading the surrounding tissue, where 3B describes the invasion of the lymph nodes in the neck. And then 4A refers to metastasis to other regions of the body and other tissues and other distal parts of the body, where 4B refers to the metastasis and invasion of the CNS, as well as brain. Lower stages are obviously more treatable and offer a higher or a better prognosis after treatment. And unfortunately, all stages will eventually result in removal of the eye to prevent spread of the tissue. And then this is the work cited. Thank you for watching.